Good afternoon, everybody. This is Paul Raj. I'm the regional head for the Vascular division within the B.Braun group, and we would like to welcome all the participants to our first Meet the Master of SeQuent Please Asia Pacific, Dr. Rosli Muhammad Ali. He is the senior consultant cardiologist of the Cardiology Department, Cardiac Vascular, Sentral, Kuala Lumpur. He's very popular in the past in the National Heart Center. He has been holding very dominant role in the EuroPCR, AICP, and also he's our founding member of the Asia Pacific drug-coated balloon group that we have here in Asia. And we have invited Dr. Rosli, who has been our pioneer user of SeQuent Please in the world, actually. He was the first man in order to start off the usage of SeQuent Please for B.Braun 13 years ago. And this is a man who knows a lot. And I'm very sure that you will also learn a lot from Dato, as much as we have over the years. And I would like to also briefly tell you what's going to happen in the next 1 hour. So Dr. Rosli will also talk about the SeQuent Please AP consensus recommendation, which we have established in 2019. He will also run through a little bit on the basket small 2, which is a very strong paper. And also he will share his de novo case sharing session. This is where the interesting part starts. And we will then adjourn for the question and answer session from 4:35 to 5 p.m., where you can, of course, either chat using the chat option, and he will be able to then answer all your queries. Yeah, and at 5, the webinar session will end, and this is how the chronology of the entire program will look like. There are some housekeeping rules which we would like everyone to adhere to. Of course, as you know, we are expecting almost 440 people so far registered for the webinar, and it's still accumulating. So we would like you to please note that your microphone has to be muted throughout the session. Please post all your questions in the Q&A section anytime throughout the session. We will then periodically inform Dr. Rosli with regards to the question that has been raised, so that he is able to also answer those questions. And the session, please take note, will be recorded for future references and also for discussion purpose in the future. So people, please bear that in mind. And without further ado, I'm going to hand over the session to Dr. Rosli in order for him to start off with the AP consensus group. Thank you, Dato, for making time for us during this season. And we really appreciate that you're going to share your session with us.
Yes, thank you very much, Raj. I would like to thank all of you who has logged in today. Which one is it? Yeah, right. You know, I find the topic rather amusing and flattering, no doubt. But I'm sure a number of you in the participants would actually be doing this procedure in more masterful manner than I am. But I'm here to share with you the Asia Pacific Consensus Group recommendations on the use of drug-coated balloon technology, at least some of the recommendations. Now, you and I know the advantages of drug-coated balloon, since it is a device where the drug is coated onto the whole balloon. It can potentially give a more homogeneous drug elution into the tissue. Since there is no stents, the risk of stent thrombosis is not there. You don't have stent struts across side branch ostia. There is a potential that you can actually use a shorter DAPT duration, and this is even in the recommendations by the Europeans. And because the fact that you do not cage the blood vessel, that you allow the blood vessel to function normally. And what is interesting is that there have been a number of papers right now to show that if you use drug-coated balloon, you find that upon restudying these patients, in this case, the first paper by Franz Kleber at 6 months, you find that a significant number of these patients actually have the mean lumen actually is bigger than post-index procedure. So you have a net mean lumen gain in the future. And this is something which is very interesting. Now, the Asia Pacific Drug-Coated Balloon Consensus Group had its first meeting together in a meeting that was organized by B.Braun in September of 2018. So a number of us from different countries in the Asia Pacific region sat down and discussed some of the things which we felt was important. And we then wanted to make a manuscript and got it published. And this is this manuscript was written by Dr. He and Professor Shin from Korea. These are some of the people that were involved, and this has been published online in Cardiology Journal. The basic and rather the background and the reasons why we feel that we should have our own Asia Pacific consensus is the fact that in Asia, we have different demographics and the patterns of disease are different. For example, we tend to have more small and diffuse coronary artery disease than in the West. The treatment for antiplatelet therapy may also differ somewhat, because if you look at a number of papers for East Asians, they tend to have a much higher risk of bleeding and with the same antiplatelet therapy as compared to the West. It is also our thoughts in writing the manuscript is that we wanted to provide adequate guidelines for the effective use of drug-coated balloon. Now, with this effective use of drug-coated balloon, we hope that this will extend the use of drug-coated balloon in the practice, especially in the Asia-Pacific region. These are some of the contents that have been written up in the manuscript. I'm not going to cover everything. I'm just going to cover what is important in this short period of time. But just to illustrate the importance of this manuscript for the Asia-Pacific region, and also for anyone who wants to use these recommendations. We know now that especially for paclitaxel drug-coated balloon, which is the ones that has the most data at this point in time, the established indication would be for in-stent restenosis for both bare metal and drug-eluting stents. But I would take a point that they have now strong data to the use of DCB for small vessel disease. And if you to look at the treatment of small vessel disease, these are lesions which are more challenging. The risk of MACE, restenosis, and TLR is much higher. And DES has been shown to be beneficial in randomized trials, but one would like to avoid stenting as much as possible because these are small vessel disease. And it's a small paper with Bellow study. DCB was shown to be better than the use of Texas over a 3-year period of time. Basket Small 2 is a significant landmark in the study of DCB to compare it versus DES in the treatment of lesions in small vessels. This is performed in Europe, 14 centers in Germany, Switzerland, and Austria. And what they did was, these patients had consent taken and they were then brought in and had PCI done. After what is deemed as a successful pre-dilatation, they were then randomized to the DCB arm or DES arm. There was no difference in terms of the target vessels or bifurcation lesions. The procedural success rate was high. The number of DCB, though, was higher than DES, even though it was not statistically significant. The length of both devices were the same. The effective size was slightly bigger in the DCB versus DES. And as you would expect it, the duration of inflation was longer in the DCB as compared to the DES arm. But suffice to show the primary endpoint at the end of the study was similar between the DCB and also the DES arm, about 7%. MACE events. When the MACE events were broken down into single components of MACE, be it cardiac death, non-fatal MI, or TVR, there was no significant difference between all the 3 arms. So they also look at the device subgroups. Now, in the beginning, the stent that was used was a Texas Liberty arm. Texas Liberty has got paclitaxel, so they wanted to compare the same drug, DCB paclitaxel versus DES paclitaxel. But as you know, over time, the Texas was made unavailable, And therefore, the study had to, protocol had to be changed, and they chose the best-in-class limus drug-eluting stent at that time, which was the Zymes. They had to recalculate the number of patients that had to be recruited to ensure that they can have a meaningful outcome or analysis at the end. So Zymes was chosen. So in this subset or device subgroups, you find that the DCB and Zymes did best as compared to patients who received Texas or who received DCB and had a conversion to drug-eluting stents. But suffice to show and to note that the outcome in DCB and Zymes was similar, and this was the same as the total study population, which includes the all-comers, and that includes also the use of Texas. So at least at this, when you look at this study, the conclusion of basket small 2 was this. It was the first large randomized controlled study comparing a DCB, in this case a paclitaxel iopamide coated DCB versus DES, and it was non-inferior to DES, and therefore small native coronary artery disease may safely be treated with DCB after successful pre-dilatation. So that leads, you know, in small vessels where you are loath to try to put stents in it, you know that if you want to use DCB, that can be an alternative therapy. Let me share with you an example of DCB in a small vessel that we performed in the past. This is a 58-year-old man. I wonder whether you can see my mouse. Okay, so the patient had a proximal LED stenosis, very severe LED stenosis, and then it was totally occluded. The diagonal was a fairly large vessel, and this was also showing significant stenosis at the same time. So we said we're going to try and see whether we can open up and see how the extent of the disease, and whether we can use DCB in a number of portions or sites of the lesion that is to be treated. We managed to cross it, pre-dilated with a 2.0 and subsequently a 2.5 balloon catheter. This was at low pressures, about 3 to 4 atmospheres, and we went up approximately at higher pressures. On the left and the right, you can see that this is the results post-balloon dilatation. The flow is very good. There's some mild dissection which is seen here, but it is non-flow limiting. So we said that we are going to try and treat the distal lesion, where a small vessel, with a drug-coated balloon. This was a 2.25 by 40 at 6 atmospheres for 150 seconds. And subsequently, in the proximal segment, we had to stent it. Even in the diagonal, we had to stent it because of significant dissection. And that is the results after. The black marks are the site that we treated with DCB, and it was overlapped a bit with drug-eluting stent, and even the diagonal had a drug-eluting stent. We restarted, this is a results post. You can see that the results are very good, and the flow is TIMI-3 flow. And we managed to restudy this patient 9 months later, and you can see that the DES site and is quite good. There is an ostial, mild to moderate ostial stenosis in the diagonal that was stented, and you can see that the vessel where it was treated with DES is patent, and you have an inkling that it is bigger than when it was as compared to the post-index procedure. So the DCB arm shows that it is larger in terms of lumen as compared to post-index procedure. Now that's for small vessel disease. Really then the treatment that a lot of people will be asking is, what about treating in patients with large artery disease? Or what we can term it as non-small artery disease, which is more than 3 centimeters, 3 mm. Obviously, this is where the cash is, because if you look at it, the interest and the treatment of these patients are large as compared to just small vessel disease. Unfortunately, the limitations with DES is that there is no robust data, and the stent data is very strong. But there have been a number of studies now to actually show us that we may actually help to have the outcome of treatment with DCB to be much better. And this is with the use of FFR guidance. There have been a number of studies that looked at a different cutoff point of 0.75, 0.8, or 0.85. But suffice to note that if you have a good FFR following post pre-dilatation, and if the results are good in keeping with angiographic assessment as well, then you may elect to use DCB. If let's say it is the cut is below the cutoff point, then one should use a DS. The group take a level of 0.75 and more for you to consider the use of DCB. Now, bifurcation lesion, also again, there is lack of strong data. There have been a number of small studies that show the benefits of DCB for bifurcation lesion, but really the treatment issues is with treating the main vessel and side branch. Do we treat both the main vessel and side branch with DCB? Or do you stent the main vessel and treat only the side branch with DCB? And again, if you choose the latter, how do you treat the DCB? Is it before or after main vessel stenting? So these are some of the issues that have not been resolved as yet, and we do need a number of large studies to actually define the use of DCB for bifurcation. But I take it upon you that DCB approach only for main vessel has two strong advantages. The first is with regards to carinal shift. You do know that and realize that once you put in a stent, especially if the stent sizing is not appropriate, you can have the carinal shift at the side branch, and this may cause a jailing of the pinching of the ostium of the side branch. And with drug-coated balloon, this can actually be avoided. The second advantage is that the side branch ostial diameter can actually enlarge over time. This is an example of OCT studies showing that the ostium of the side branch at baseline, post-balloon, and at 3 months, there is an enlargement of the ostial diameter. So these are two potential advantages if you can even consider DCB for the main vessel. The other important aspect that was in content was with regards to what is considered, or we're giving guidelines to what should be considered as an optimal lesion preparation for both in-stent restenosis and de novo lesion. And we took a bit of space to actually write this down. The European guidelines based on the German's guidelines or recommendation is that if you are to consider DCB only strategy, lesion preparation is important. You pre-dilate it with a semi-compliant balloon, inflation pressure, which is a bit more than nominal, with a balloon to vessel ratio of 0.8 to 1. Obviously, in complex lesion, you can think about non-compliant balloon, arthrectomy, cutting balloons, and so on and so forth. If you get a result post pre-dilatation, which is an acceptable angiographic results with no major dissection, it means say that there's either no dissection or dissection grade A, type A or B, with good TIMI3 flow and restenosis of less than 30%, then one should be able to consider DCB. When you choose a DCB, the balloon to vessel ratio is recommended at 0.8 to 1, and you should have, should just inflate a nominal pressure for at least 30 seconds. Now, if you do not get acceptable angiographic results, then you should put it on drug-eluting stent. Our own manuscript mentioned that coronary dissection with TIMI3 flow or with uncomplicated or non-flow limiting dissection has a favorable outcome and predicts a low restenosis rate. We also remind the readers is that DCB is only designed to deliver an antiproliferative drug. It is not meant to be like a drug, meant to be like a normal regular balloon angioplasty. The balloon itself is not as deliverable as a normal regular balloon, and you should not be using it to inflate at high pressure to overcome the stenosis. You should do it before you place in a DCB. So really the key to successful DCB treatment depends on whether the lesion preparation is appropriately and adequately performed. Now, obviously, if you have non-flow, if it's a flow limiting dissection, you should use DS. If a non-flow limiting dissection, you can consider DCB. And in this case, if you have got an FFR, and you can actually use FFR in terms of guiding your therapy. There is a paper that was published just about 2 years ago with regards to optimal lesion preparation, in this case for in-stent restenosis. They looked at 3 parameters. The first is to look at the diameter stenosis of the lesion preparation, and they take the cutoff point of 20%. If it's less than 20% as compared to more than 20%, the outcome is much better at 3 years as compared at 2 years as compared to patients who had residual stenosis of more than 20%. Secondly, they also look at a factor of DEB or DCB to stent ratio. Now, the German consensus was 0.8 to 1. In this case, they put that it should be more than 0.9. So you're actually using a much bigger balloon. And that has also shown to be favorable. And thirdly, they look at the total inflation time of DCB. When you have a longer inflation time, then you find that it is more than 60 seconds. It is better than less than 60 seconds. So all these 3 factors. And if you get all these 3 parameters obtained, then the results is comparable with the 2nd generation DS. So this is for in-stent restenosis, and I believe that this can be replicated for de novo lesions. I am going to share with you 2 patients with regards to vessel preparation, and this is, I feel, an important area. The first is a 62-year-old gentleman whom I have been treating since 1997. He had inferior STEMI with familial dyslipidemia, extremely high cholesterol. We had PCI in 1997 to the RCA, stented there. LCX in 2002 also stented there, and that was with bare metal stent. He came back in 2015 with distal RCA ISR that was treated with TCB, and we stented right from the prox ostium to the distal RCA because there has been a progression of disease. Now he came back again, and we find that, and upon restudying, we find that there's now severe stenosis in the LED, not much of calcification, and this was the results in the right. The right is still very good. You can see that the PD is occluded, and I decided not to treat this lesion at all because it's going to be more complicated, and we treated just the LED lesion. The other lesion that has also been found was a severe stenosis in the proximal OM, high OM, and this was quite a significant size balloon. We prepared the vessel, and this was treated with a scoring balloon, 3.0 by 13, and that's the results of the post-scoring balloon. And you can see that in the short segment, there is a dissection. This is probably a class C dissection because the contrast, there's some contrast hold up in this area, but the flow is very good. So here it is. The question would be, can you stent it or not? Recommendations, of course, C, you should stent it, though it's not hard and fast rule. So in this case, I really wanted to try to avoid stenting in this patient. So I waited for about 5 minutes, and I took a picture, and the picture, the images was the same. You still have that contrast being held up, but the flow is all right, and there is no residual stenosis of less than 30%. So I elected to use DCB, and this was with a 3.0 by 40 mm at 10 atmospheres for 90 seconds. And that was the results after. So once again, if you're not sure, you need to wait, because sometimes over time, the dissection can become worse, then you have to convert this to a drug-eluting stent. And this is where it's good. If you have another lesion, you treat the other lesion first and you wait. So in this case, the high OM was treated next with a POBA. And then that was result post-POBA. And we find that acceptable. And we treated this with a DCB 2.25 by 15. And this was the results after. You can see that results shows fairly good, POBA results with in the OM. There's dissection noted in the OM, but this is a type B dissection. It goes away and the flow is very good. So I accepted this. And this is after 15 minutes post-drug-coated balloon. So you find that if after 5 minutes, the flow is still very good, the chances of abrupt closure is going to be very low. And this is the results in the LED. You can see that the results are still the same. There is still this stain being held up. Residual stenosis is not significant. and the flow is very good. So I accepted this, and this was 30 minutes after post DCB in the LED. We managed to, this is another results again. Once again, it's good results with TIMI3, well, almost TIMI3. We decided to accept this and brought him back to be restudied in 7 months. And once you can see that the DCB in the OM is much better than post-index. And that was results in the LED. The LED dissection has actually healed and the flow is preserved. And I just want to show you the flow post-procedure. So this is what you will expect if you prepare the vessel very well, choose the patients that you decide should be suitable for DCB. Because otherwise, if you are not happy with it, of course, you need to stent. And if you're not, if you choose the patients wrongly, then the conversion rate from DCB to DS may be high in your practice. So another patient with a 76-year-old man with severe calcification in the arteries, and the right coronary artery is severely calcified, severe stenosis from the ostium and until the mid-segment. We treated this with the orbital diamond-back atherectomy system and then stenting. And you can see the results are good. We brought him back 2 weeks later to treat the left side. There's severe calcification. The distal left main is significantly diseased. There's also disease in the significant stenosis in the ostium of the circumflex. This is another view to show the stenosis in the LED. And you can see that the severe stenosis, another severe stenosis in a mid-LED. And this is another view of the lesions that we are going to tackle. So we treated once again with orbital atherectomy. And this is the results post OAS. The flow is poor, but we haven't treated adequately the proximal. We then took to prepare the vessel better. We used a scoring balloon, the Scoreflex, from mid until proximal. And you can see the results post, and especially the mid segment looks fairly good with hardly any significant dissection seen. The proximal was treated or prepared better with a 3.0 non-compliant balloon till about 20 atmospheres. And that's the results after. So in this case, you can see clearly that there's no dissection that is obviously seen in this segment. And we said that we are going to plan for DCB from just proximal to D2 to enter the mid segment. The rest of lesions we wanted to stent, especially the left mid. This was first treated, the mid segment with the SeQuent 2.5 by 30, and in the proximal 3.0 by 25. And then we stented the other lesions. And that was the results after. You can see that the flow down in the LED was good. And in fact, the DCB treated site was also good and pleasing. Okay, so when it comes to vessel preparation, especially in complex lesions, I think one needs to be, to try and prepare the vessel very well with an aim of getting less than 30% residual stenosis, less than 30% residual stenosis post FOBA with a good TIMI3 flow with no major dissection. You may require, in this case, atherectomy devices to get rid of and modify the calcified plaque, and also in the use of scoring balloon or non-compliant balloon at high pressure. And as often, that is a teaching, you should not get any significant waste post vessel preparation, and therefore you don't have significant residual stenosis. These are the patients when you use that DCB, you can have an enhanced penetration of the drug into the vessel wall. So to summarize, ladies and gentlemen, DCB treatment is certainly an attractive therapeutic option. It has a number of advantages over stenting, but obviously you need to have confidence in using DCB and that it works. Because when you use DCB and you want to, you believe in the technology and you want to use it as much as possible, there has to be a change in your practice. You need to be more patient. You need to have much better vessel preparation than DS, for example. But with the possibility that you would probably see more dissections, but at least you need to be aware that you should not be afraid of all dissections. Type A and type B, they are rather safe, and risk of the blood closure is very low. And you just need to practice and practice using DCB until you get it right, because you want to limit crossover stenting. Because if you have a high crossover stent rate, then that would defer you from using DCB in your patients. So I'd like to say that it is an attractive concept of treating lesions with leaving nothing behind. So thank you very much, ladies and gentlemen.